Hello everyone, this video has been created to help you address some of the past paper questions that have come up on the AQA A-level biology papers for enzymes and some of the required practical 1 questions as well. I've added this document onto my test shop for free, so you can download it, attempt it yourself. The mark scheme for these questions are always on the back of that document, so remember these downloads are absolutely free for you. You may as well just kind of have a go at doing them and then come to this video to watch some of the explanations for some of the questions. So the first question number 1 that I want to go through is some of the basics. So for 2 marks, the question asks you to define what an enzyme is. So the first thing that I would say is that an enzyme is a protein. You could always go on to say about how enzymes are biological catalysts also. Both of these points would give you 2 marks, but there are some other points that you could make. You can see here that they do accept you to say that enzymes will speed up a reaction instead of saying that it's a catalyst. So you could get a potential 2nd mark for that. But other points that you could make to talk about how the reaction between an enzyme involves a substrate as well, or a specific substrate, and how enzymes can also lower activation energy. So there's at least 4 points that you could say for those 2 marks. Nice and easy so far, I think. 1B asks you a 3-mark question, and it basically just shows you a diagram of 3 stages between these different shapes that are labelled up as D, E, F, and G. And it says that this is an enzyme-catalysed reaction, and it asks you for 3 marks to describe what is happening in this reaction. You do have to, for 3 marks, make reference to the names of the shapes, and as part of your 3 marks, you have to identify which shape is an enzyme, which shape is a substrate, and which shapes are the products. Now, if we look at stage 1, we've got shape D and we've got shape E, and stage 2 shows shape E fitting into shape D. So that should give you an indication that D is the enzyme and E is the substrate. So for the first couple of marks, they expect you to kind of say that enzyme D binds or collides with substrate E, and talking about the active site changing shape or forms an enzyme substrate complex with the E. Now, if you talk about induced fit, then that would get you an additional mark as part of that. If you don't make any reference to the letters, then the maximum number of marks that you could get in this particular question is 2. So that's something to kind of be aware of. And then the last kind of mark or any other points that you might be able to talk about is talking about an enzyme substrate complex forming and then saying that the products are F and G. So these are the 2 separate shapes that it's given you. And then the last point you could say is that the enzyme remains unchanged at the end. We already know that as part of enzyme-catalysed reactions. Question number 2 gives you a graph, and it talks about the investigation that's taken place on the effect of temperature on the rate of an enzyme-controlled reaction. And they've tested 2 temperatures, 25 degrees, which is represented by the dotted line, and the solid line, which is 37 degrees. And you can see that both of those shapes of those graphs are slightly different. So the first part of the question asks you about a factor that the technician would have controlled. Now, we know that he's testing temperature, so temperature is not going to be one that they're controlling. In fact, the temperature is going to be as part of the independent variable in this particular experiment. You are changing that, so that's not controlled. So the factors that we would be looking at are different factors that would affect enzyme activity. So you could mention concentration of substrate solution or concentration of the enzyme, and you could mention pH as part of that as well. So that's an easy 1 mark to obtain. So for part B, they ask you to calculate the rate of reaction at 25 degrees C. So you're looking at change in X and change in Y. And the answer that you should have got for this is 2.5. And then the 2nd mark is given to the unit. So it should be grams per decimeter cubed for that one. And then I think these questions are really important to understand. The describe and explain in terms of the command word. So this one says describe and explain the differences between the 2 curves. So remember, describe is to say what you see, and explain is to say why you think that that's what's happened as part of the experiment. So we just need to pick one of the temperatures. So for the mark scheme here, they've looked at 37 degrees. So in order to describe what's happening at 37 degrees, you could split it up into two different parts. So the first mark goes for the initial rate of reaction that's faster at 37. You can see the gradient of that graph is steeper, therefore the reaction is faster. And then as part of that explanation, well, why is it faster? Well, because it's a higher temperature, therefore there is more kinetic energy. And where there is more kinetic energy, there are more enzyme substrate complexes formed, or more collisions that occur. And then you just say the second part of the graph where there is a plateau at 37 degrees, that this particular temperature, there is a plateau there that is reached. And then we explain why that plateau takes place. So at this particular point, the substrate's all been used up, so the rate of reaction remains at a standstill, effectively. Say, for example, you've chosen 25 degrees to describe and explain that, then it's just the converse, that the rate of reaction is pretty steady all the way from the beginning of the experiment to the end. There is no plateau because the substrate is not completely used up as part of that. And the rate of reaction is slower at 25 because there is less kinetic energy in the molecules, and so there are less enzyme substrate complexes formed in the same time when compared to the 37-degree graph. So skipping forward to question 8, this particular question asks you to describe why an enzyme catalyzes only one reaction. So what you need to talk about is, say the enzymes have an active site. Remember that active site is a word that you have to mention. It's underlined as part of the mark scheme, so you have to talk about the fact that enzymes have an active site and how the substrate will fit the active site. Note that on the mark scheme, it will reject you saying that the active site is the same shape as the substrate. It's not the same shape, it's a complementary shape. And there are students who might also say that the active site is on the substrate, so you can't say that, you wouldn't get the mark for that. Question 8b actually talks about competitive and non-competitive inhibitors. So that's the first thing that you need to be able to recognise as part of this question. It talks about gout being a disease caused by the buildup of uric acid crystals in the joints and how uric acid is produced from xanthine in a reaction that's catalysed by an enzyme known as xanthine oxidase. It talks about a drug called allopurinol, which is a drug that's used to treat gout, and it shows the structures of xanthine and allopurinol. So the first part of the question for 3 marks says, use the information to suggest how allopurinol can be used to treat gout. So we would talk about how allopurinol is a similar shape to xanthine. It's both got 2 rings of those nitrogens, it's got the NH bonds, and it's got that double bond with the oxygen. If you said that it's the same shape, well, they would reject that because it's not identical. If you said similar structure, obviously that's fine, that's what the mark scheme expects you to say. For the second marking point, you could talk about how allopurinol is a drug. Because it's used to treat gout, you know that this is going to enter the active site of the xanthine oxidase. And if it enters the active site of the xanthine oxidase, then it's going to be a competitive inhibitor. So any one of those 2 points would get you that particular second mark. Note that for the second mark, if you talked about enzyme substrate complexes, you wouldn't get that mark associated with that. And you also cannot say that it's a non-competitive inhibitor, especially if you know that it's taking up the active site, that it's not going to be a non-competitive inhibitor. And then for the third mark, if you've said already that allopurinol is a competitive inhibitor, then you know that less xanthine is going to bind with the enzyme to then create the uric acid. Something along that point would be really useful to say. So less xanthine binds, there's fewer enzyme substrate complexes that are formed, and therefore there's less uric acid, and so gout is treated. As part of the third mark, if you said there's no enzyme substrate complexes formed, you wouldn't get it. So you've got to make sure that you say less. Then fast forwarding to question 10b, it says scientists have investigated the effects of competitive and non-competitive inhibitors of the enzyme maltase. And the question actually asks you to say, describe competitive and non-competitive inhibition of an enzyme. Now this is worth 5 marks. So essentially what it's asking you to do is to say how competitive and non-competitive inhibition works. So there's a lot of points that can go on to say this, and I'll put them all up on the screen and we'll talk through them as we go through. So first of all, you could talk about what inhibitors are as a definition. from my video, you know that inhibitors reduce the binding of the enzyme to the substrate. They can stop a reaction from taking place. They can prevent the enzyme-substrate complex from forming as well. So you would get 1 mark for saying that. And then you can talk about competitive and non-competitive. So if you're going to talk about competitive inhibition, then you need to say that with competitive inhibitors, they are a similar shape to the substrate, and they actually take up the active site so that the substrate cannot bind to the enzyme. And we also know that this type of inhibition can be overcome by adding more substrate. So there's 3 potential marks that you could get for mentioning about how competitive inhibition works. For the non-competitive inhibition, we know that the inhibitor will bind on the enzyme but not on the active site. So saying something like that would get you 1 mark. You'd also then need to talk about how the active site changes shape so that the substrate does not fit. So any of those points in the combination would get you the 5 marks for this question. Hopefully that makes a bit more sense now that you've seen an extended response answer. Okay folks, that's all I've got for you on that. Please remember that the document for these past paper questions is on my test shop, and you can access that using the link that's just below this particular video. If you click on it, it will take you to that. It is free for you to download. So please complete those questions. There are 10 questions on there. I've only gone through a small portion of those questions, but of course the mark scheme is attached onto that document, so you shouldn't have any problems with that. Thank you so much for watching and joining Bioteacher on this video. Bye for now.